Okay, welcome everyone to the 15th annual CompuCell modeling workshop. I'm going to start out with a brief review of CompuCell and some of the things that it can do. We post all of the links in the chat as we go along. I need to remind you that the meeting is being live streamed on YouTube and it's being recorded. So please keep that in mind as we go forward. I'm going to quickly go over the course logistics. Some of you have just heard that, but for people who are just joining us, I'll do that. I'll remind you about the hackathon and on resources, and then we'll start actually talking about CompuCell and modeling more generally. Logistics. You have the Zoom links here. The same Zoom link that you used today should work throughout the week. There'll be a separate Zoom link for the hackathon. Please do make sure that you can register for those. We can repost the hackathon link in a second on the chat. If you're going to do the hackathon, we ask you to put your idea into the hackathon document, which is shared in Google Drive. The Slack also has a hackathon channel, and there's a folder where people with ideas should upload a slide where they can come and present them tomorrow. Zoom logistics, please keep yourself muted during the presentations and ask questions through the chat or through the Slack channels. Feel free to use the go slower, go faster buttons. Feel free to use the raise hand button. All of the exercises and slides and videos will be made available afterwards if you miss something. We don't want to leave people behind. But sometimes things happen and you have to miss a session, and so we'll do our best to support people who can't be here the whole time. Again, you've heard me before. I'm James Glazier, Indiana University. We have TJ Sigo, Gilberto Tomas, Giulio Belmonte, Giuliano Ferrari Gianlupi, Jim Sluca, and Josh Ponte Serrano, all of whom are going to be giving presentations. Bobby Matamanchi may be helping out, and we have volunteer help from Professor Lorenzo Veschini, King's College London, and Pedro Castel from Brazil. We're very grateful to them for their help. The daily schedule will be, for those of you who were here earlier, an informal orientation to the day's agenda and question session from about 10 a.m. to 10:30. We'll start formally at 10:30 a.m. till noon, short break. Then the second session of the day, a slightly longer break, which we call a lunch break, but what time zone you're in will depend. Make that maybe something different, like a dinner break or a breakfast break. And then we'll have 2 more modules followed by an opportunity for more discussion and troubleshooting at the end of the day. The tentative course outline today, we're going to be introducing CompuCell and the basics. I'll be lecturing for the first hour and a half, then Giulio will take over in the late afternoon. Tomorrow, Giuliano will start again. Then we have those hackathon lightning talks and possible discussion about teaming. TJ will show some of his COVID modeling work, and then we'll introduce chemical fields. Wednesday will be focused on the interactions between cells and chemical fields and begin to discuss modeling subcellular networks. Thursday will mainly be devoted to modeling subcellular networks and their control of cells. And Friday will be discussing more advanced topics like links, solids, and compartmental cells. And then next Saturday and Sunday will be the hackathon. Again, I'm going to put the hackathon poll up one more time. I know some people have answered this already. If you're just trying to get a rough sense of how many people want to participate. And it's fine if you change your mind as you see how things go. You don't have to have an idea to participate. But we definitely encourage you. In the past, it's been a lot of fun. and people have come up with some projects that have led to PhD theses, publications, and so on. If you do have an idea, please reach out to us. If you want help with an idea, please also reach out to us, and we'll be happy to work with you. Does anybody else want to state an opinion before I close the poll?
Yeah, I press submit and it doesn't work twice. So, but I intend to be there observing Saturday, Sunday.
Okay. Well, I apologize. Is anybody else having trouble using the poll? Okay. Okay, but it looks like we have plenty of people who would like to participate. So that should be fun. Look forward to working with you on that. Again, if you have a hackathon idea, you can post it in the Slack channel. Please do add a brief description to the shared Google Doc, and please upload a single PowerPoint slide on your hackathon idea, ideally by tonight at midnight. If you absolutely need 2 slides, that's okay, but also make sure that your name is in the slide so that when we go through them, we know who to call to present. Okay. Resources. We have 150 people registered for the course. We have 80 people plus on the call today. And so that's a lot of people. We have a good staff, but it's certainly possible that we could have a little bit of overload. So if for some reason you need extra help, please don't hesitate to raise your hand. We have breakout rooms where we can have people give you one-on-one assistance. Again, resources, the CompuCell website, the student materials folder on the Google Drive. I want to draw your attention again to the Tellurium Manuals. There are also some very good videos on Tellurium that are available from Herbert's course 2 weeks ago. I especially call your attention to the Slack and to the quick reference guides. Those quick reference guides are really helpful. There's a couple of page guide to Python that has everything you need and more. There's a great quick reference guide to CompuCell3D that will remind you of all the things that we cover in the class and some things we won't get to. And for subcellular network modeling, there is a very good guide to Tellurium that in one column of text gives you everything you need to know. And so I strongly recommend printing those out and having them available, or at least having them bookmarked so that you can find them. If you want to learn more, there are a couple of other documents in the student materials folder talking about how CompuCell works internally. And there was a nice paper by a Canadian group of students on some of the general issues about modeling in computational mathematical biology that we thought was worth taking a look at, although it's not specifically focused on what we're going to do for this class. Now I'd like to start the lecture proper. The first thing that one might come to is asking the question, why bother with this kind of model? And you can come to that kind of model from many different points of view. One of the things that one might think about is that there are needs for novel approaches in medicine. If you want to be able to personalize medicine, if you want to be able to predict what's going to happen to a patient when you treat them in a particular way, you need to have models of how those patients behave. And there's a whole set of concepts around the idea of medical digital twins that are developing very rapidly. All of those are built around the idea of being able to build predictive quantitative models of complex biological processes. And so one of the key things that you need to be able to do is to be able to predict. And this course will be focused on building predictive models, not predictive models typically of individual patients, although that's certainly something that one would like to be able to do eventually. But in our case, more asking the question, how do tissues organize during development? How do they maintain themselves once you've built the organism? How do developmental diseases cause disorganization of tissues? Because we'll find that cancer and many other developmental diseases have very great similarities with normal tissue development. We're going to talk a little bit about infectious diseases, how hosts and pathogens interact, and one can use these methods also in bioengineering, in particular tissue engineering, to try to understand how to repair tissues or how to build engineered tissues. If we look especially at early embryonic development, we find that there is an enormous amount of movement in tissues as they develop. And one of the reasons that we developed CompuCell in the first place was to be able to understand and model that kind of cell movement. And in fact, CompuCell has been used to model intersegmental vessel growth in zebrafish, branching morphogenesis, and segmentation. And so these are the kinds of deployments that CompuCell was initially developed for and has been used for pretty successfully. There are a lot of similarities, and I'm not going to go into detail in any of these slides, between developmental mechanisms, maintenance mechanisms, and the mechanisms that come into play in cancer, how tissues are maintained, how cells control their proliferation, how cell death is controlled are all things that are of importance in all three of those contexts. The difference between stem cells and somatic cells and differentiation are also important in all of those contexts. And those are things that you can build CompuCell models to understand. Tissue organization is also something that we're going to discuss a bit. The initial things we're going to do with CompuCell are going to be mesenchymal cells. That's the natural, simplest kind of cell. But we're going to show you how to build models of epithelial cells, that is, polarized cells that form layered structures, and to understand how those work. The extracellular environment is going to be important as well to how tissues organize. CompuCell does not do a lot of extracellular solid modeling, but it is good at modeling extracellular chemical fields, and you can implement some ECM solids also with it, although that's not something we're going to talk about in great detail today. TJ has done a good deal of work on building quasi-solid models using this kind of methodology. One of the things that motivated a project like CompuCell is that there isn't a clear mapping between genes and cell behaviors. If you ask the question, for example, what genes cause cancer, beyond P53 and one or two other genes, there are very few genes that are mutated in most cancers, because cancer is really a disease of cell behavior. It's cells behaving in a way that they shouldn't in the context they find themselves in. And there usually are many molecular ways of doing the same thing. And so really what matters very often is not so much specific molecules, but rather cell behaviors. And CompuCell is going to be built around the idea that cell behaviors are critical to understanding how tissues are built, maintained, and how they fail. More generally, quantitative modeling is important because biology is full of what are called as feedback and what are called incoherent feed-forward networks. So if I imagine that molecular species A excites the production of B, promotes the production of B, and promotes the production of C, B promotes the production of D, but C inhibits D, and I increase A, I don't know from looking at that diagram whether increasing A increases or decreases D. Similarly, if I have a cell that's migrating on a substrate, say agar, and I change the stiffness of the substrate, I don't a priori know whether the cell is going to move faster or slower. And so this kind of modeling that we're going to do is really critical when it's very difficult to extrapolate from molecular signatures to cell and tissue behaviors. The particular question that you're going to work on in any given case will determine the level of detail, spatial and otherwise, that you need. And it's always worth asking the question, is it worth building a complicated model? Sometimes very simple models are sufficient. And that would come up a lot, for example, in some disease models. And so as you're working, especially if you're going to do the hackathon, you should be asking the question for yourself, what is the simplest model that I could build that would give me the answers that I want? What is it that I want to understand? What are virtual tissues? Virtual tissues are the kinds of simulations that we're going to build in this class. Typically, they start with some kind of concept of subcellular regulation. That could be a network or a logical diagram or something else. A set of cell behaviors that we're going to implement using the cellular POTS model GGH method that CompuCell uses. And then an extracellular layer of chemicals that diffuse, solids that stay in the environment and influence how cells move, and so on. And this is a simulation you'll see again and again. It's a somewhat old simulation of segmentation in a chick embryo. Each one of the cells on the right is running a very complex subcellular network of oscillators. Those oscillations are in turn controlled by an extracellular gradient of a morphogen, in this case FGF8. When that morphogen reaches a critical level, the cells differentiate depending on the oscillator and then change their mechanical behaviors that leads to segmentation. These are mechanistic, dynamic, agent-based multiscale models, and our focus is going to be on spatiotemporal structure and behaviors. They're mechanism-driven, they're not data-driven, and they're additive. Nothing exists in your model unless you assert it. Not even the statement that cells have a volume exists unless you assert it. And they're going to be based on quantitative models of how objects behave. You'll have models of how cells are born or created, how they die, what forces they create, how they change shape, how they move. And the typical output of these models is movies. And then, of course, you'll also do analysis of those time series to get numerical results that you can compare to experiments. One dream that we've had for a long time is that you could take the molecular detail in static images, so-called tissue atlas data, use those as initial conditions for simulations, and animate the dead tissue to predict what would have happened. And that's something that we're working on quite a bit. And if people are interested in discussing that more later, we'd love to talk about it. These are not easy problems to work on. Biology is hard. There are lots of components. There are lots of component types. There are lots of parameters. We don't always know how things work. Biology is intrinsically noisy and stochastic. Individual components of the same type may not be the same in detail. Even the same cell will behave differently when I change its environment. All of our models are phenomenological. None of them are going to have all the details in them. And sometimes comparing experiment and simulation isn't so easy. Because, for example, if I grow a vascular tree, each time I grow that vascular tree, that network will be different. How do I compare a random network of blood vessels produced by a simulation to one from an experiment? Those are things that are still open scientific problems in many areas. And so there are a lot of things that we have to address when we build these models, but that's not a counsel of despair. It means there's a lot to do, and it can be rather enjoyable to do those things. So now I'd like to get everybody up and running with CompuCell. Just to begin with, I want to do a very, very simple exercise. Please, everybody, go to the CompuCell tool on NanoHub. That's the CC3D base 4X tool. And if somebody could post that in the chat for me, that would be great. Okay. And I'll use my poll to ask everybody able to get into NanoHub. If you're using the desktop app, that's fine. Click yes, that's fine too. Okay, it looks like everybody pretty much is getting there. I'll wait another minute or two to make sure that it's working for people. If you're on this screen, you hit Launch Tool, and it'll ask me to log in. And in my case, you can sign in with Google if you're not in an institution that they recognize, or you can create an account here. I can sign in with Indiana University, and it knows who I am, so it'll go up for me. Once you've gotten there, you want to do, if you haven't done it before, NanoHub uses something called the dashboard. To get to the dashboard, you can either go to dashboard here, you can hover up here and get dashboard, or go up to the top right where it says logged in and select dashboard. And then you can go to all tools. And if you haven't done it already, you can look for CompuCell. It'll give you a variety of options. The one we're going to use here is this CompuCell V4 main tool, and hit the little heart here to bookmark it so that it is available to you. And then hit the window here to launch it. And so now I'd like to double check and make sure that everybody can get CompuCell to launch. And occasionally CompuCell is buggy online. And if it gets buggy, I recommend hitting terminate and relaunch. Okay, so everybody seems to be okay on that. And our first exercise will be running a very simple simulation. CompuCell comes packaged with a variety of demonstrations, some of which we'll use. You'll also be writing your own code. CompuCell has two main tools. CompuCell Player, which is what you're seeing on the screen now, which is for executing and simulations and controlling them, and Twidit++, which is a model editor where you're going to develop and change the simulations. Okay. You're inside of NanoHub, you're going to be able to resize your window. You can change the size of the viewing windows as well. You can move the boundaries of your boxes around. Something that may happen to you when you're using CompuCell online is that NanoHub doesn't always redraw the screen properly. And if you get a messy screen, if you grab that bottom right-hand corner and joggle it a little bit, if it'll let me do it. Sometimes it can be annoying as well. It will redraw the screen for you there. So if you get a messed up screen, I recommend resizing a little bit, okay? So to open a simulation, we can go to File. I'm sorry, I'm running the code, so I have to stop it. It's one problem. I tried it out this morning and I had it set in the wrong configuration. So go to File and go to Open Simulation at the top. And click on your username. And you should find something which is called CompuCell3D, CompuCell demos here. I have to find it. It's you should have a simpler directory structure than I do because I have a lot of these. CompuCell3D demos. Inside of CompuCell3D demos, there should be a simulation called, let's see, did I go to the wrong place? Demos again, extra layer of demos, and then book chapters, and then angiogenesis. Does everybody have that hierarchy? It's listed here on the screen. Okay, somebody says that they need some help. So if I could ask our assistants to contact the person who needs help and move them into a breakout room if necessary. If you need help, please use the chat or raise your hand using the Zoom, and we'll get you the help you need. Okay. Try that again on the poll just to make sure that everybody's caught up. So the full path for the demo. It may look slightly different in your computer. Click on your username. And then once you get to your username, you should find a folder called CompuCell3D_demos. Inside of demos, there is a folder called demos. Inside of that folder, there's something called book chapter demos. And inside that folder is something called angiogenesis. And again, if people need help, we'll come back to this. So if it's a problem, you'll have plenty of chance to practice. So don't worry too much. Select angiogenesis.cc3d and hit open. And once you've started that, you've now got that file loaded. And you'll see a set of 4 buttons at the top of your player. This round circle is step. Ooh, bug. That's interesting. Let me try that again. Well, TJ, I seem to have a problem with running my own demo. I'm going to do what I suggested, which is hit terminate and relaunch. Did anybody else have a problem with that? Everybody else had it crash as well? Oh, did I open? There we go. Open file. Demos. Chapter. Angiogenesis. Well, this isn't going to be the best start to the course if we can't get this to run. I'll try one more time, and if I can't do it, I'm going to turn it over to the team to get it working, and I'll move ahead to the rest of the lecture. Let's see, last time. CompuCell main tool. Launch. Open simulation. Click on my username. CompuCell3D demos. Demos, book chapter, angiogenesis, angio. Open. There we go. So who else can run? Was ever people able? People seem to say mine runs, but doesn't show video. It runs. It runs. Third rerun. Okay. Somebody TJ says it may be a problem with NanoHub service. That's possible.
Yeah, so this happens sometimes, especially when a lot of people sign in all at once. So if we just hang in there, probably everyone with the desktop app isn't seeing this issue, and it should resolve itself here pretty quickly.
Okay.
May I ask which one? Is this the angiogenesis or the other longer name?
Angiogenesis.cc3D.
Okay.
So somebody asked the question, legitimate question, how many people is too many people? And the answer is once everybody is in, it should be fine because NanoHub has a lot of servers, but it's possible that if too many people try to load the file at the same time, it's having a problem. So let me try again, now that it seems to be working for me. It's definitely was not a problem with the package. So let's see, how's it going? Okay, again, if people need help, we can move you to a breakout room if you identify yourself. What you see on the screen may vary a little bit. I want to emphasize that there are 4 buttons. The first button is run, which will run the simulation. The second button is a single step, which will advance the simulation 1 step and then wait. The third button is a pause button, which pauses and then lets you do a single step or run again. And the last button with the square is stop, and stop will reset the simulation to the beginning and start over. Okay. You should see a simulation window that looks like this. And there are a couple of things that you want to learn about it. One is how to resize it. You can drag it, resize it using the usual buttons. There's a very helpful tool, which is if I go up under window and I hit the second option, which is tile, that will lay out the screen in a way that makes sense. A third thing that you might want to know is that you can display in 2 dimensions or 3 dimensions. You probably started, in your case, in a 2-dimensional simulation. If you want to see in 3D, you select 3D. So why don't people try that? And then you can pan and rotate your simulation. Occasionally, the simulation will get lost. If you hit R while you're hovering over the simulation, it should recenter it and bring it back. So why doesn't everybody select 3D and hit play and see the simulation run? Okay, again, are the people who are who need help raising their hands or otherwise connecting with our assistants to do the help? Okay, so you now have a running simulation of neoangiogenesis. Again, if you get the simulation off in a funny situation, you can hit R to recenter and restore it to its original value. And you can use the mouse roller to zoom in. You can move over the mouse over there to change the view and so on. So why don't people just take a second or two to try that out? Let me look at the chat. Somebody says they're running on the desktop and it works. Zoom in, zoom out is your roller bar on your mouse, or you can use the zoom button here, zoom in, zoom out. One of the things about the CompuCell interfaces is that there are a lot of different options for doing the same thing. And so I tend to use the same ones over and over again, but you may prefer something else. There are a lot of shortcuts and options for doing these things. And so you have to decide which ones are the ones that are most comfortable for you. How are we doing in terms of getting people getting the exercise working? I'm going to move ahead a little bit. If people need help getting the exercise working, please reach out to one of our instructors through the chat. Okay, and I'll read Paul in just a second. Okay. CompuCell offers a lot of visualization options. Those visualization options can be accessed here. Depending on whether you're in 2D or 3D, you may want to render the cells, the cell borders. If you have subcellular elements, cluster borders, and a variety of other options there. I'm not going to play with those very much at the moment, but there are plenty of things you can play with. The other thing that you can do is this rather cryptic icon that's called configuration here will allow you to change various settings. There you can ask your simulation to save images and save movies using the output option. You can set a little bit about how the player is configured under general settings. We don't use that one so much. If I don't like blue as the color of my cells under cell color, I can select a different color for it. Maybe since they're vascular, I'll make them red. We haven't looked at the chemical fields yet. In 3D, sometimes you want to have some of your cell types be invisible so you can see inside of a 3D object. You can set that here under 3D. And then there are various other kinds of axis settings, which we're not going to worry about so much here. Again, one of the things you'll find in CompuCell is that there are an awful lot of options, and it takes some time to explore them. One of the things that you're going to face here, you might try changing the color of your cells here, if you like, as we're talking. One of the things that this particular simulation has of blood vessels is that blood vessel growth depends on a growth factor. It's called vascular endothelial growth factor, VEGF-A, and there's a chemical field. So if I go under here, this button here, it says new graphics window, or if I pull down on window here, new graphics window, I can create a second graphics window. Why doesn't everybody follow along and do that? And I can say that instead of seeing the cells, I want to see the chemical concentration. And now I have to relay out my screen because I've hidden part of my screen. I go to window and I hit tile. And here I have my vascular endothelial growth factor field on the left and my cell field on my right. Generally, when you do a major change in the way the fields that cells are displayed, the simulation will pause and I have to hit run again to continue the simulation. So why doesn't everybody just take a second or two to play with that a little bit and see what happens? You can do a 3D visualization of the field, although that's a little bit more complicated to interpret. That's going to give you isosurfaces of the chemical concentration. So is everybody able to get that to work? People still having problems? And you can see that these endothelial cells have organized to form a three-dimensional network of capillaries. Again, I see people saying needing help. If you need help, please do reach out and ask for help. Somebody says, I can't see any output. My window is black. Is the person who said that running on a desktop machine? Okay, yes, you do have to do a tile usually to make sure that things are well behaved. And again, if you hover over a window and you hit R, that should recenter things. Sometimes when I change the cell border and color and rerun the simulation, the cell color remains the same in the screenshot. Okay, yes, Giuliano. Sometimes you will actually get a simulation to disappear. There is a bug that's possible. To reset the rotation of the 3D visualizations so that both have the same orientation. That's a good question, and I believe it's possible to do with text, but I don't think there's any way to scroll both of them simultaneously. So you'd have to reset the orientation and then do the scroll separately in the two. Good point. Sometimes when I change the cell border, color and border and rerun the simulation, The color doesn't change. Can't see both graphs at the same time. Try to use the layout, the tile. Can you see the initial conditions? Pedro, if you're having a crash, make sure you're running the copy that's in your user directory and not the copy that's in the CompuCell root directory. Let me show you the initial conditions. If you want to see the initial conditions, hit stop and then hit step, the single step. Okay, so that's the initial condition. Here, a cube of cells and no chemical field. And if I step one at a time, those cells begin to secrete vascular endothelial growth factor and they begin to reorganize. And if I were going to reorganize things to try to make the configurations match, I would do it now probably. And have to play around with it a little bit to try to get them oriented in the same way. That's not too bad, I think. And now I can run. Again, this was just a first exercise. I understand that some people may have an issue. I'm going to monitor the chat here. When you change the color setting, you have to remember to hit OK after you've changed the color, or it won't save it. There, it didn't change. So I guess that's a problem I'm having as well. That's interesting. Haven't seen that one before. Let me try it again. Change the color. Endothelial cell. Color. There. All right. So one of the excitements of this course is that you never know what's going to happen, including when I run things. And so I appreciate your patience when these things happen. Server disconnected. That one I will have to refer you to one of our team to help you out with. Okay, I'll let that run. And now I want to talk a little bit more about CompuCell. Again, in the next module, we'll be doing a lot more about these things and we'll have plenty of time to get any bugs worked out. One of the key things that we see in this simulation is that we have cells, individual cells. Let me go into 2D and show the cells. Maybe zoom in a little bit so we can see them better. The borders are not very easy to see, so let me change the border color. Let's see, cell border color. Instead of yellow, let's make it black so it's clear. And here we see the individual cells, and we see these cells are composed of pixels with boundaries. And if we're going to describe what they're doing, we have to describe what the configuration of the cells is, what forces are acting on them, causing them to move, and the rules for how they move. And there are a lot of different ways of doing this. There's a rather old book now that talks about some of these that you might want to look at. You can represent cells as points. Some people may have taken Paul Macklin's class last week. Those are point-like cells. These are called center model simulations. There are many different modeling environments that use center models. There also are cells which have explicit shapes. CompuCell is an example of that. There are other simulation environments like Morpheus, Virtual Leaf, that use the POTS model algorithm. There are so-called vertex models that are used especially for modeling things like fruit fly development. And they're more finite element-like models which have very explicit mechanical descriptions of cell surfaces. those models tend to be very detailed, computationally expensive, but also very productive if you want to understand biophysics of tissues. If you want to learn a little bit more about the methodologies in CompuCell, there's a nice book from a few years ago by Shanna and Preziosi that actually shows the simulation we're running here. The simulation is the one here on the cover of that book. Again, CompuCell represents cells on a lattice. You'll hear more about that in a few minutes. Each cell consists of multiple voxels or pixels. And so it's a little bit like pointing a camera at the cells and saying that every voxel in cell number 1 is number 1, every voxel in cell number 2 is number 2. CompuCell has a lot of core ideas about how cells behave and what they do. Remember that these are constructive models. Nothing exists unless you assert it. And so cells have to be given things like volume, surface area, viscosity, elasticity, polarity, their behaviors like cell movement, cell growth, cell death, and their interactions like cells sticking to each other, responding to chemical fields, differentiating, secreting chemicals, absorbing chemicals. Their mechanical structures called links that we'll talk about on the last day of the class that allow you to build quasi-solid materials. Their chemical fields, we're seeing one here on the right. In the case of VEGF, those can diffuse and decay. They can also react. And there can be subcellular networks that model signaling and regulation and control. In this particular simulation, there are no subcellular networks. The specific numerics of how these are done are things that Julio is going to talk about a little bit. When you model things like cell adhesion, you have to imagine that each surface unit, here a yellow cell is touching a green cell, has an energy associated with it. And so you'll see a term called contact energy that describes how cells stick together. Mathematically, that's written as a sum over the cell lattice with particular restrictions. Cells have volumes. You can't actually specify the volume of a cell exactly, because if I imagine, for example, suppose I had 100 balloons. If I have those balloons sitting out on the table, each balloon will have its own volume. But if I squeeze those 100 balloons into a box, it's a small box, they'll shrink because they'll be under pressure.
Hello again, James. I think you're back.
I'm back. Is my screen share working?
No, your screen share is missing.
Okay. Well, if this may be good, because if all the bugs happen in this first lecture, then maybe there won't be any more bugs for the rest of the class. So one of the things that we'll learn quickly is that we specify a lot of cell properties in the form of what are called elastic constraints. In this case, we say that the cell wants to have a volume of V target, and there's a penalty associated with that cell not having its target volume. And again, this is something that Julia will come back to later in the day. The way in which things move is a little bit complex. If you haven't had physics, these are based on some properties of what's called statistical mechanics. CompuCell is what's called a stochastic model. At each time step, micro time step within a simulation, it looks at the boundaries of each cell. It tries to move randomly the boundaries forward and backwards. It asks the question, does the energy of the system change? That is there a force on that boundary? And it makes that move with a probability that's associated with a fluctuation amplitude. The details we're not going to go into here. The key thing to know is that the result is that boundaries move with a velocity that's proportional to the force. And in the subcellular world that we're working in, there's almost no inertia. If you stop pushing on something, the boundaries stop moving. And so you want to be in what's called the overdamped limit, where the velocity is proportional to the force. And the details of this you'll hear about a little later. This method breaks down if I push too hard. And what you'll see then, if you push too hard, is that your cells begin to fragment and become dust-like. And that's a particular pathology of this modeling method that we'll come back to. And now for the rest of the lecture, I'd like to talk a little bit about some of the kinds of questions you can answer with CompuCell. We're a little bit behind in terms of my timing, but we still have half an hour to go over some things that you can do with it. These are questions that we've asked over the years in our group, but there are many others that have been asked. Everything from why do blood vessels sometimes invade the retina in age-related macular degeneration, to could we personalize treatments for diabetic retinopathy, to why do some people have a lethal cystic outgrowth in their kidneys, to why does acetaminophen sometimes cause liver necrosis. Those all seem very diverse. They're all problems that have been addressed with CompuCell models of one sort or another. If we go back a long way, one of the very first simulations that was done was based on experiments where you mix two cell types and you ask the question, how do the cells reorganize when they have particular surface adhesion properties? And that's a simulation we used to use as our first simulation. These days we tend to avoid it, but if you want to run the cell sorting simulation as a demo while I'm talking, you can do that. You can find that simulation in those demo directories. Clearly, we need cells to be able to grow and divide. This is a simulation of somatic evolution in a simplified tumor derived from the work of Heiko Enderling, who's now the head of the Society for Mathematical Biology. In this case, the yellow cells represent stem cells. The red cells represent somatic cancer cells. You see those cells growing and dividing and filling the space that's available to them. That's very easy to implement with a target volume, because you could have the target volume of the cells increase and then have them divide. And you'll hear about that tomorrow. I've already shown you the idea of a chemical field. In this case, in this middle column, the cells in red are secreting a chemical. That chemical is diffusing and decaying in the extracellular environment and leading to the formation of a gradient of that chemical concentration, which is the color scheme that you see here in this middle row. Cells can also respond to chemicals by chasing the chemical, by moving up or down gradient. That's called chemotaxis. This is a classic simulation where we have simulated leukocytes, which are chasing bacteria. The blue cells would represent leukocytes. The green cells would be bacteria. The bacteria secrete a chemical that the leukocytes recognize and chase. That chemotaxis is also happening here in the angiogenesis simulation. These cells that we've shown here in the angiogenesis simulation are very simple. They're just blobs of material, but you could build more complex simulations where the cells have detail. This is a simulation of a crawling cell where the cell has a nucleus here in gray, a lamellipodium in green, and a cytoplasm, which is made invisible so that you can see the nuclear and lamellipodial compartments. You could also model chemical movement within the cells, and Giulio's done a lot of work on that. I've mentioned the possibility of modeling epithelial tissues, layered tissues, and so you could use subcellular compartments to define apical, basal, and lateral surfaces of cells. Giulio has taken that to quite elaborate lengths, modeling, for example, the origin of polarity in cells through chemical signaling within the cell. And so people who are interested in modeling what's going on inside of cells are able to do that as well. Here's another one of Giulio's simulations, in this case showing the patterning of hair cells in a Drosophila imaginal wing disc. This is again a simulation where the individual molecular species inside the cell and their interactions across the cell boundaries are modeled explicitly. I just showed that simulation of a crawling cell. One of the things that's quite interesting about cells is they have very specific statistical behaviors when they crawl. They do what are called persistent random walks. And that's a simulation that you'll learn how to write and run. And one of the things you could do is say, if a single cell behaves in a particular way, what happens when you have multiple cells interacting with each other? And Professor Tomas from Brazil will show you how to do that. This is the simulation that we just ran in the 3D version on the right. This is actually an old experiment by Ambrosi et al. Showing human umbilical vein endothelial cells organizing to form a vascular pattern. And this was old work that we did trying to understand what specific mechanisms the cells needed to have in order to be able to make a vascular network. In this case, the simulation comes in two flavors. The cells respond to the chemical by chemotaxing, moving up gradients of the chemical. In this case, on the right, the contacts between cells, the regions where the cells are touching each other, don't respond to the chemical, and they form a network. On the left, all surfaces of the cell respond and they form droplets instead. And that's a simulation we could actually run very easily here on the right with a single line change in the code. You could take those blood vessel networks and have them act as sources of nutrients for cells. And this is a simulation of a vascular tumor where an initial tumor cell in the middle receives nutrients from the blood vessels. It proliferates. As the tumor grows, the cells in the middle of the tumor become nutrient deprived. They first become quiescent and then necrotic. And you'll see that there's a necrotic core in this simulation. In the first case, those cells that are quiescent and don't have enough oxygen send out a growth factor that causes proliferation of blood vessels. Those are the purple lines here. In the second case that they don't, the blood vessels stay put. They're still pushed around by the growing tumor, but they don't proliferate. In both cases, the tumor grows, but the pattern of tumor growth is quite different. And you can understand the interaction between blood vessels and tumors using models of that kind. This was a simulation of evolutionary effects in tumors. Cancers are notoriously prone for developing resistance to chemotherapies and radiation. During the progress of cancer, you have what are called the hallmarks of cancer as cells become more motile, less responsive to signals for inhibition of cell proliferation. And this is a simulation of the evolution of those behaviors in a very simplified way. One of the things that I mentioned is that we can try to do things that are more, these are very abstract models. We can try to build models that are more medically relevant. Your retina has a very complex architecture. At the back of the retina is a thick membrane called Brooks membrane here in gray. Behind it are the choriocapillaris, the blood vessels that supply the majority of oxygen and glucose to the retina and get rid of most of the waste products. When you have a retinal scan, the blood vessels that you see are only a fraction of the blood vessels that actually supply the retina. In front of the Brooks membrane, you have the black pigmented retinal epithelial cells that line the eye. And then on top of them, you have the photoreceptors. And that's the basic structure of the retina. In a certain number of people, especially older adults, although also in people who get eye infections or who have trauma to the eye, mechanical trauma to the eye, these blood vessels, the choriocapillaris, cross Brooks membrane and begin to proliferate inside of the retina. And when they do that, you can go blind within a period of 6 months to a year. And this was work that we did a number of years ago, trying to understand the mechanisms behind that. At the time, there were no good treatments for this problem. And we built a simulation of the retina. Here are those in gray are those Brooks membrane. The purple cells are the blood vessels behind the retina. Green are the pigmented retinal cells. And here in dark gray are the photoreceptors. There's a small hole in Brooks membrane. The blood vessels penetrate into the retina. They begin to proliferate in the retina. And as they proliferate, they spread, and they eventually will cause retinal detachment. There. And the breakup of the retina with the loss of vision. This was a simulation where we actually calibrated the parameters pretty carefully. And so the number of days here represents real time, and the simulation is able to replicate the time scale over which this is observed to happen in real individuals. And one of the things that was quite interesting was we were able to investigate what happens if you in fact change the adhesion of the pigmented cells to Brooks membrane, of the pigmented cells to each other, of the pigmented cells to the photoreceptors. Each one of those causes a problem in the retina, but a different problem. And we were able to build what's called a risk map that identifies what the likely effect of a particular mechanical defect at the cell level is at the system level. As you age, inevitably, lipids build up on Brooks membrane. It's not replaced during your life. And that means that the pigmented retinal epithelial cells stick less strongly to Brooks membrane than they would otherwise. It's one of the reasons older people are more likely to get retinal detachments. And that increase of lipid with aging is the primary risk factor here in red for this kind of retinal invasion and breakup. And so that actually gave some ideas about new ways of approaching treatment of diabetic retinopathy. I say diabetic retinopathy, age-related macular degeneration associated choroidal neovascularization. You've seen this simulation of segmentation before. Again, this is a model with detailed subcellular oscillator, model of cell differentiation, and a model of extracellular chemicals. Giulio Belmonte, in his PhD thesis, worked with a group in London that discovered that you could get the same thing to happen even if you didn't have the chemical oscillators or the gradient. And so Giulio worked to build a mechanical model of cell polarization and reorganization of epithelia that was able to explain why you were able actually to get this kind of segmental patterning even in the absence of those oscillatory states. And that's a simulation that is a little bit complicated, but something if somebody wanted to work on in a hackathon, Julia might be able to help out with that. In this case, you have cells that have compartments. You have an apical surface here in green, basal surface in red, mediolateral surface in blue. The cells elongate. These lines are those links that I talked about earlier. In green here is extracellular material between the cells. And you can build then these kinds of reorganization models. This is another model of the same kind of process done by Priyam, who was a graduate student here, who's now postdoc at Duke, looking at the organization of an epithelial layer, what's called apical contraction at the top of the cells, leading to the failure of the epithelial layer and the formation of clusters. Another thing that we've worked on from a more medical point of view is something called autosomal dominant polycystic kidney disease. It's a genetic disease. At the moment, it's treatable only with kidney transplant. It's associated with a mutation in two genes, polycystin 1 and 2, but exactly how those gene mutations translated into the cystic phenotype wasn't understood. Our experimental collaborators, clinician, Dr. Bob Bacayao, who's a surgeon, kidney surgeon at our medical school, hypothesized that it was due to an adhesion defect where the adhesion between the cells in the kidney was not appropriate. This is a simulation of a small section of kidney tubule. We've introduced a single cell which has a different cadherin in it, a different cell adhesion molecule in it from the normal one. That leads to interference with contact inhibition of proliferation, the signaling that tells the tissue that it's intact. So this is a normal tissue repair mechanism that is getting misdeployed. That failure to respect that signal to say, don't divide, leads to inappropriate cell division and the formation of cysts. We were able to test various hypotheses about how that missing information played out at the tissue level. This was an experiment done using a kidney cell line, which had been transfected with this ectopic cadherin, this inappropriate cell adhesion molecule. What you see is that the cells in green are the ones that have the wrong cell adhesion molecule, and they're forming cysts. And that gave us some insight that we've used to try to develop drug therapies to treat this disease. Giulio will be showing you a simulation of what's called convergent extension. This is a process that occurs early in development in many animals, where tissues shrink along one direction and extend along another direction. This is simulating the filopodial contraction of cells, here in 2 dimensions, here in 3 dimensions. And that's a simulation you'll do later in the week. Another thing that we could simulate, and this is a new simulation, is signaling between cells using a pair of cell adhesion molecules called delta and notch. The delta and notch form are what are called a heterotypic pair. Delta on cell 1 binds to notch on cell 2. Notch then gets cleaved, and the intracellular component of notch sends a signal. That signal in turn leads to cell proliferation, and you get a particular kind of patterning of the tissue where cells are either high delta or high notch, and the proliferation of the cells also then depends on the delta notch levels. That's another simulation that you'll learn how to build and operate later in the week. One of the things that everybody has done a lot of in the past year and a half is modeling of viruses and immune response. This is a detailed model that TJ built of a patch of lung tissue. The lung tissue has an initial infection with a virus, in this case modeling influenza virus. The infected cells send out a variety of chemical signals. Those chemical signals lead to the invasion of the tissue by a variety of immune cells. There are quite a few here. There are neutrophils, CD4 plus T cells, B cells, antigen-presenting cells, natural killer cells, and CD8 plus T cells. So quite a few cell types. And there also is quite a bit of signaling going on. There are TNF alpha, a variety of interleukins, a reactive oxygen species, which the immune cells use to kill infected cells. There's type 1 interferon, type 2 interferon, and antibodies. And in this case, you're actually able to calibrate this simulation against very detailed data for influenza infection in mouse lung, and able to reproduce the difference between low-level infection, a low dose of infection early on, and a high dose of infection, leading to different outcomes in the mouse. You can use that kind of modeling also to model drug therapies. These were some preliminary work we did last year, modeling remdesivir therapy. In this case, remdesivir inhibits the replication of viral RNA when the virus has gotten into a cell. In that simulation, every single cell had its own model of viral replication. You can also have your own model of interferon-induced antiviral resistance, and you're able to simulate what happens if you give the drug, in this case, the blocker of RNA synthesis, at different times after infection. At the left, we give it at the same time you're infected. At the right, as we go right, we give it later and later times. At the top, we give a very high dose of the drug. At the bottom, we give a low dose. And the coloring essentially represents whether the treatment worked or not. These lines represent the number of uninfected cells that are present in the simulation. If the line goes down, it means that all the cells in that patch of lung tissue are becoming infected. That's bad. And we're able then to come up with a result that tells us that, unsurprisingly, the earlier you treat, the better. The stronger you treat, the better. Those aren't too surprising. But the shape of the boundary between those two regions is rather interesting. And you also see, if you zoomed in a little bit, that there are many regions in which some patches have successful treatment and some don't. One of the things that I mentioned early on is that biology is highly stochastic. And so we have to deal with the fact that our simulations represent one replica of potentially many, many, many different outcomes. And so one thing that we'll do later in the class is teach you how to do replica simulations and parameter scans to try to understand the details of the stochasticity. An area that we've worked in quite a bit more recently is diabetic retinopathy. It's an increasingly frequent cause of adult blindness. Again, like with age-related macular degeneration, there aren't really very good treatments for it. The main way that it's treated these days, first I should say, what happens is that the capillaries now on the front of the retina become tortuous, and they start getting leaky, and they also die. And that leads to dropout, that is, patches of the retina that die. And the main way it's treated today is by going in with the laser and actually killing the photoreceptors, which need oxygen. And this is on the principle of forest firefighting, where you cut down trees to try to prevent the fire from spreading. The problem at the moment is that the way the trees are cut down, or in this case, the way the photoreceptors are wiped out, is essentially unintelligent. You see essentially a random or almost grid-like spot pattern. And so what we tried to do was calculate where there was likely to be a problem in the retina and suggest that you should try killing the cells where there was likely to be a problem, so that you should build your fire break where it was needed, not in random locations. And if you do that, you can reduce the area of damage from the treatment itself, and also reduce the risk of spread of the vascular damage. That's a project I'd love to get back to. It got a little bit thrown off course by COVID. And so if somebody were interested in working in that, that's something we could talk about as well. Another thing that we've worked on a lot is liver toxicity. Tylenol or acetaminophen is a very common headache medication, prescribed quite a bit. It has the unfortunate effect that it can be very toxic to the liver. In fact, it can cause death from liver necrosis. And that's unfortunately rather common. One of the things that's a bit mysterious about acetaminophen is somebody can take it safely for a long period of time and then have a low-level overdose that's lethal. We spent a number of years trying to understand the details of that. These were simulations where we were trying to match experimentally measured blood levels versus time of acetaminophen and its metabolites to a detailed simulation of flow in the liver, trying to understand which cells are exposed, why they're exposed, why they die, and to build a more detailed model of how that toxicity happened. So that was a really lightning run through of a variety of problems that we've worked with over the years with CompuCell. Again, all of those were CompuCell models from our group. CompuCell has been used by a lot of groups over worldwide to build models. I don't mean to suggest that The list of things that I've shown you are by any means exhaustive. There are plenty more things one could do with it. But I hope it gives you a sense of some of the possibilities that you can achieve using CompuCell. I also realized that showing one slide about cancer evolution or one slide about tumor vasculature isn't very satisfying. For each one of those slides, I would give a whole hour lecture on the details of the biology behind it, how the models are built, how they're validated, and what they mean. But I thought to introduce the course, I'd like to come up with a list of just possibilities and some pretty movies that would allow people to have a sense of some of the possibilities of the things that you could do with CompuCell going forward. Ultimately, CompuCell has been mostly used for understanding the basic biology of development, tissue maintenance, that is homeostasis and developmental diseases, and more recently, infectious diseases. It's been used also to predict effects of chemicals and drugs, both for toxicity and for treatment. And I showed some examples of that. And in the future, we'd like to be able to design new therapies. That polycystic kidney disease example is one, where we did come up with some drug targets based on those simulations. And ultimately, we'd like to be able to build personalized therapies. We'd like to be able to build models that actually represent not only generic cases, but specific individual cases. And I think in the case of tissue engineering, that's going to happen fairly soon. And this kind of modeling, again, I started out by saying you have to ask what kinds of questions you want to answer before you decide what kinds of models you're going to use. All of these models involve spatial effects. Things are not the same from one region of the tissue to another. If things are all the same, you can do simpler models that are a lot faster to run. And most of them involve cell movement. And so almost everything we're going to show you about simulation during this course will involve cells moving. In most of your body, cells move. Depending on the tissue, that may be something that happens all the time, or it may be something that happens with only a subset of the cells. It may be something that only happens, for example, when cells die and have to be replaced. There are a few tissues where you have relatively little cell movement. In the cornea of your eye, there's almost no cell movement. In muscle, cells don't move relative to each other very much during normal function, but when myofibers die, the remaining myofibers reorganize to take up the missing space and fill in for the cells that have died. That's specific to humans. There are other animals in which muscle fibers are replaced continually during life. So I hope that gives you a little bit of a sense of the capabilities of CompuCell. I apologize for that snafu. Can I ask people, I'll poll, I'll do one more poll. I'll do a quick poll. I'll relaunch that first poll. How many people were ultimately able to get the things to work for that simulation that I showed? If there was anybody who was not able to get it to work, please do reach out during the break to one of the team for help. And I want to end the module by asking you for your feedback. And I appreciate your patience with the problem with the can people see that poll? There we go. The problem with the NanoHub when we all tried to log in at once. I said we have a very large group. And so we have a bigger group today than before. And so we never, I think, had 70 people try to log into NanoHub all at once. So I think that that's a problem. Once you're logged in, you shouldn't have any more problems. And so I hope that that's not going to be a snafu going forward. So please fill out this end of module questionnaire. I really appreciate your feedback on that. I'll give people a few minutes to do that. If there are any questions that you'd like to put in the chat now while we're doing the questionnaire, feel free to post them. And let me know if there's anything I can do to help out. Okay, with that, I'll leave the questionnaire up for another minute or two. We're scheduled for a 15-minute break. When we come back, Giuliano is going to be taking over, and we'll have plenty of practice actually getting the NanoHub demos to work better than I guess I was able to do. So I thank you for your participation in the first module. I hope things get smoother as we go forward from there. If anybody else has any comments or questions, please let us know. And please do fill out the module questionnaire. We'd love to have your feedback. I'll leave it up for another minute. Team, is there anything else that we need to go over before we take our break?
I'm just gonna ask people to download a zip file that I'm gonna send the link now, so that I can teach people how to upload CC3D simulations to NanoHub.
Okay. So is that going to be in the student materials?
Yes, it's
Google
there,
it.
and I've sent the link straight to the file as well.
Okay, so if people want to take the time over the break, Giuliano will have a file to download. One of the issues that you'll find with NanoHub is that uploading and downloading takes a couple of steps. It only takes about a minute to do once you're familiar with it, but we do have to show you the steps. Okay, well, thank you, everybody. Appreciate your patience again.